Hi all, thank you very much for joining us for this webinar. I'm going to share some slides and give you all the information for today. All right, so today we're going to talk about the work that we have been doing here at TileDB for a long time in genomics. The title of the webinar is kind of provoking, and I will back it up. We're saying that population genomics is, well, currently a data management problem. It's a very challenging problem in general, but we feel that currently it is kind of being blocked and slowed down. And I'm going to give you all the information about this today. So I will start by presenting who we are and how we came here and why we're working in genomics anyway. TileDB itself is a research project that I had started when I was working at MIT and Intel Labs between 2014 and 2017. It has deep roots at the intersection of high-performance computing, databases, and data science. So high-performance computing because of Intel, databases because of the folks that I was working with at MIT, and data science because we were extremely fortunate to talk to a lot of scientists and practitioners across the domain. So we got a lot of insights from multiple different applications. I was the original developer of Genomics DB. Some of you guys may have used it. It's part of GTK4, and that was a collaboration between some great people at Intel and the Broad Institute. It was a collaboration. A lot of good people worked on this, but that was the very first use case of TileDB. So that's why this is quite sentimental for me and very, very personal. That's when we realized that a solution like TileDB is worth doubling down on, and that's what led to the creation of the company. We are well capitalized, we're VC backed. That allows us to take decisions very, very fast and do build things without any strings attached. So that again, that allows us to be very, very aggressive. Currently, we're 40 members. And we have expertise across all applications. For example, my colleague Aaron today, he's a bioinformatician. So when we work in specific verticals, we partner with people that know the domain very, very well. And we have had quite the traction with telecoms, pharmas, hospitals, and other scientific organizations. TileDB is coined as universal. There is a reason for that. But again, today is all about genomics, and I'm going to tell you why TileDB is extremely important for genomics. A disclaimer that I always include in my webinars: all the credit for all the amazing work you're going to see here goes to our powerful team. I'm just a mere representative, so check it out at tiledb.com/about. And also please know that I'm the exclusive recipient of complaints. So if something doesn't sound right to you, please send me an email. Before I even begin with the agenda, I would like to give some special thanks to an amazing partner that we've had over the years, and this is Helix. We have, first of all, their mission is to empower every person to improve their life through DNA. They're doing amazing stuff at that company. We have had a multi-year collaboration with them to scale population genomics workflows. They provided tons of ideas and optimizations to the TileDB-VCF offering I'm going to explain today. So I wanted to take the time to thank them. Please find more info about Helix at helix.com. All right, so this is what we're going to do today. First, I'm going to describe as carefully as I can the problem that I see in population genomics. Please know that I'm a computer scientist, all right, and an expert in data management and databases. I am not a bioinformatician, so I'm going to give you perhaps a different perspective, and everything I'm going to say is going to be unfiltered and very pragmatic. Okay, so hopefully this is going to give a couple of insights from somebody that is coming from other domains, bringing knowledge from many other applications. Then I will present a general solution. I'm not going to overpitch TileDB. I will explain what TileDB does, and of course we have a section where we're going to show you how this works. But for those that are going to set about to build their own solution, that's all fine. But I would like to share at least our insights when we build this solution. So I am going to give you a framework for a general solution here, and then I will present a concrete solution. So consider TileDB, at least for today, as an instantiation of this general solution to this problem. And then I'm super humbled and very excited and honored that Dr. Stephen Kingsmore is joining us today. he's a giant in genetics. I'm not gonna do him justice if I present his background. I'm pretty sure that most of you on this webinar know him very well. He's going to present his vision and very bold project on genome-informed inpatient pediatric care. And this is one of the more gratifying aspects of our work at TileDB. We get to work with brilliant people like Dr. Kingsmore, and we try to contribute as well in the way we can to important projects for humanity. So this is extremely important to us. Then I'm going to hand it over. So we'll try to keep this within reason in terms of time, because then I would like to allocate sufficient time for Dr. Aaron Olin, my colleague, to walk you through the actual solution. So I will present the theory, but Aaron is going to dive deep into the technology and actually show it to you in action. And finally, I'm going to conclude with some work in progress, and I do have one last insight to share there when it comes to work in progress and building for the future. Okay, so let me start with the problem. And on this slide, I mentioned that this is just a problem on the surface, but this is what is blocking pretty much everybody. But this is just the beginning of your problems. I'm referring to the bioinformatician folks on this webinar, whoever is watching this. So I will be brief because I assume that most of you coming here have the background knowledge. So population genomic analysis is performed on these files that contain information about samples. Let's say 1 VCF file per sample. In this particular case, I'm going to make it simple. And these are large collections. Could be millions in the future, hopefully billions. 1 per person, could be 1 per cell. Like, let's say 1 per person in this particular case. The analysis is done by slicing a portion across files. So, for example, the analysis may be focusing in a particular region within a gene per sample. So this means that the analysis needs a portion of those files across the files. And there are mainly 2 approaches. First, you keep separate single sample VCF files. That's all fine. So you need to access every single one of them and get this piece of information. Or you combine those VCF files into a single one, which is called a combined VCF file or a population VCF file. Okay, and I'm going to explain the issues with those 2 approaches soon. But then the domain uses specialized downstream tools. They expect the VCF format, they expect VCF files. And most of the solutions we've seen are around these files and also building environments, building the specialized downstream tools and so on and so forth. So all the work in genomics we've seen revolves around these VCF files and those specialized tools and building environments for bioinformaticians to work in. So what is the problem with the single sample VCF approach? So the latency from accessing every single file may be minimal in your eyes, maybe 1 millisecond. It's never 1 millisecond. You may be storing these files on S3, right? And the latency there is larger. But in any case, even if it is 1 millisecond, if you have 1 million files, this latency adds up and it is killing your performance. It makes it non-practical at all. So to mitigate these problems, the domain adopted solutions that combine these files into a single one, because in that single file, the information across the files gets to be collocated, be very close to each other in the file, and therefore with a single sequential access, you can get a lot of the relevant information to the query, to the analysis, very fast. So that solves the problem with the single sample VCFs. The problem is that the storage space of this combined file scales super linearly with the number of samples. What this means is that if each VCF sample is 500 megabytes, and you have 1,000 of those and you combine them, the outcome is not going to be 500 gigabytes. So 500 times 1,000, 500 megabytes times 1,000. It's going to be a big multiple of this. The storage space explodes, and there is a reason for that. I'm not going to cover it today, but this is a fact, and it is a well-known fact in the domain. And of course, in addition to the storage, also the generation time for those files increases super linearly. Even worse, the multi-sample VCF files cannot be updated. If you want to add one more sample, you need to completely destroy that file and rebuild it. And this is very problematic, because those data sets are very rarely studied. So the side effects are that scaling population genomics is completely blocked. So everybody is focusing on solving this problem because these will not scale. And even if you have today a solution that works with 10,000 samples, it's not going to work for 100,000 samples. It's not going to work for 1 million or 1 billion. So what we're looking to do is target at infinite scale, find a solution that scales indefinitely. But most importantly, and something that I'm going to back up in the following slides, is that this is just a tiny fraction of the problem that you guys have, right? I mean, this is the immediate and the obvious one, but this is just a tiny fraction. Once you solve this, there are potentially even more important problems that will follow. And this is what I would like to summarize in this slide. So if you try to take a look at the holistic problem in population genomics, number one, I don't see any data management around. I don't see principled data management in the sense or in the way we're doing it in other domains, in the way that we have been doing it in relational databases for so long. I don't see that in population genomics. And the best way to explain this perhaps is not in the context even of data management, because when somebody thinks of data management or databases, they're thinking of SQL, and this is a very wrong way to think about this. What I would like to explain this is in the context of holistic data economics for the genomics space, because probably this is going to resonate a little bit better. So let me explain what I mean by that, and we're going to take each of those problems apart one by one. So by data economics, I mean effectively three things: the production, distribution, and consumption of genomics data. This is what I mean. So for production, we're talking about what format is the data stored in and where is it stored. So for example, the source, the sequencing company, where do they store this data? What format do they export the data in order to get them ready for consumption? Distribution has to do with who has access to the data, the governance, right? Who is allowed to see the data? How are they accessing the data? And also, is there any monetization around this? And don't be alarmed. Monetization doesn't necessarily mean that you're going to be selling patients' data. It may be, you may be selling insights. You may be selling derivative results from the analysis. So it's not just the sensitive data, right? So distribution deals with all of that. And finally, consumption, and this is dealing with how we're going to access the data. What are the tools? Where does the computation happen? What kind of computation? So the VCF problem I mentioned in the previous slides completely ignores production and distribution, completely, because people are hung up on the VCF files. That's it. It's de facto. You know, the data is going to come in VCF files. So the production problem completely ignored, and then distribution is far away in their minds, only because they cannot even make sense of the data. The volume is so big that distribution, we need to think about it later. Like we first need to be able to analyze this data. So in our mind, we need to look at the problem holistically from production to distribution to consumption. So again, let me give you more details, and I'm going to tailor this to genomics a little bit better, hopefully resonating with a couple of you folks. So let's talk a little bit about the production problem. So once again, what you guys have is a bunch of VCF files, that's de facto, and also a couple of tables. There are always tables storing phenotypes, for example, or any other clinical data. So this is the input, right? And then you're storing this data in some kind of storage cloud storage, and this is because it's cheaper, so it's very, very logical to do that. And these data sets are going to get out of hand, or some kind of a file manager, right? So that you say, okay, this person has access to this file, but everything file-based. Now, what happens is that afterwards, the domain takes specialized applications, specialized for VCF, for example, and there is a lot of wrangling and fusion because the specialized solutions may not be dealing with tabular data, for example, in the way that databases do. So there is a lot of wrangling, a lot of fusion of the data. And finally, there has to be some analytics infrastructure, like some machines, resources, scalability, and all of that. Now, because of the production problem, because of these VCFs, this wrangling over here is extremely slow, extremely expensive. And also this infrastructure is most often custom built. Every organization builds a different infrastructure that does the same thing. It analyzes VCFs. And so it is very expensive to build and very time consuming. And this is because the data came in a format that is not analysis ready. It's not ready for analysis. We need to wrangle. We need to build specialized tools to access this kind of data. Now let's move to the distribution problem. Again, the data comes in VCFs and tables. Then we're storing this data in some cloud bucket, again, or marketplace in case you do adopt solutions like this. And then each of the organizations accessing the same data, either public or private, they have to download the data. Every single one of them, replicate terabytes of data, download the data, and then do the same thing that I mentioned before, wrangle, build analytics infrastructure, and so on and so forth. So the problem here is that there is massive reinvention because everybody's building the same infrastructure. And of course, the downloading is very expensive. There is another distribution problem if we flip a little bit the scenario. For example, again, we have the VCFs. We're storing everything at some data owner. So some data owner is bold and says, I'm going to consolidate the data. I'm going to offer the data to everybody else. So I'm going to build the infrastructure. I'm going to do the wrangling. I'm going to take up all of this effort. And then the rest of the people can just start asking queries. So this is great, but here is the problem. The data owner bears, first of all, the building cost, and there might be reinvention across all the data owners, but also the data owner bears the distribution cost as well. Now, think about situations where you guys have built solutions like this using grants. So you build the solution with the grant, and now you need to maintain it in order to give access to so many other organizations. The other organizations are paying nothing, and you're paying the cost. So you need to get another grant just to maintain this infrastructure. And more often than not, one of the organizations querying the data monopolizes the whole infrastructure. Some query this infrastructure a little bit, but others are doing much more work. So this is not good economics. This cannot scale for that data owner, right? And some data owners may just not want to build this infrastructure, but at the same time, they may have some very valuable data that they really want to share with others. Finally, the consumption problem. Again, the VCFs, storage in some cloud bucket or server, and then within an organization, right? I'm going back to the original VCF problem. Within the organization, what we have seen is that we have talked to different groups accessing the same data. They do the same things like download, wrangle, copy, all of that stuff. They use different tools. But then what happens is that this is very inefficient, and there is very poor governance, because once you download the file from this server, the server is protected, right? It's a file system, it's a file manager, there are some access policies, but then you download the data locally, right? From that point onwards, there's no governance, there's no accountability, there are no insights to draw, because you don't know what this user is doing. You don't know which genes it is operating on. You don't know what insights are trying to get. So you have no information. So the consumption, this complements the consumption problem that I mentioned in the beginning. All right, so what are we going to do? And this is the generic solution that I mentioned in the beginning of my slides. First, folks, it's high time you moved on from VCF. That format is not analysis ready, and it is creating huge headaches for analysis. You need to adopt from the source, from those that are running the pipelines producing those files. Don't store your products in VCF files. Choose a format which is open source, of course, but it should be universal and it should be analysis ready. This means that it is accessible by any tool. It can be cloud native. You don't need to download something like these files in order to access them. You can just slice them. There are going to be no downloads or copies. It's going to be efficient. It's going to solve the N+1 problem. So you need something that is open to updates, that enables fast updates as new samples are coming in, and something that can unify even the tables, right? Because if you choose something for variants, but that something is super different from tables, you will still end up having those wrangling and fusion problems that I mentioned. So extra care on what you choose to store your original data at the production level. Now, at the distribution and analysis level, you need a data management platform, not a file system. You need to depart from file system solutions. So it has to be one infrastructure. You need to be able to own the data. The data is yours. It's the most valuable asset that you have. You own the data. But at the same time, this infrastructure should be responsible for global scale governance so that you can share the data with others, so that you can collaborate with reproducibility, so that you can even sell data or code. You may want to sell your code that you're building on this infrastructure. And also, equally importantly, the cost finally needs to be shifted to the consumer. The more a consumer consumes, the more they must pay. And as far as the consumers go, they should be free to use any tool. I mean, in your organizations, you're hiring people with data science skills. They may be using Python, they may be using R, they may be using the next big thing in data science. You need to let them. You cannot tell them to use the specialized legacy tools that you have been using for years. Technology advances. So all data science tools, and there should be no infrastructure hassles. The data scientists are not interested in building data engineering infrastructure. Most of the data scientists we are interacting with, they have switched into a data engineering role, and this is not productive for the organization. So this is the holistic solution. It may sound like a stretch, but let's see a particular example with what we did with TileDB. So as far as the format is concerned, what we do, and I'm going to tell you how, is that we're converting those VCFs into an analysis-ready universal format, which is the multidimensional arrays. I will dedicate one slide to explain this and show you the actual solution for population genomics. Then we build an actual universal data management platform where you can unify your variants, your tables, any other clinical data in a single platform. And this comes with all the governance that you want, right? For who has access, which portions of the data they have access to. And this is scalable and serverless because, again, the data scientist should not be acting as a data engineer. They're data scientists. And this allows you to share data and code and even monetize this work. And we have adopted the pay-as-you-go model, which shifts the cost to the consumer. If you're a data owner that wants to distribute the data to others, first, you don't need to build the infrastructure. The infrastructure is done by TileDB. And second, you don't need to pay beyond your storage costs because we want you to own your data. We don't host the data. You do, you just register it to the platform, and then the platform takes care of everything else. But then even for the consumers, you can use any tool at any scale. So TileDB comes with extreme interoperability. Now, this opens the door to the entire data science ecosystem, not just the genomics software ecosystem. And you don't need to handle infrastructure anymore. The infrastructure is done by TileDB. So that's the solution. So how do we achieve this? And this is getting slightly technical. The next two slides are going to be the only technical slides I have, I promise, because then I'm going to let Aaron explain how everything works. But this is the model, guys. So we adopt this very mathematical structure, very foundational. It's called the multidimensional array. It comes in two flavors, dense and sparse, depending on whether you're storing values for all the elements of the array or not. For VCF, we will be focusing on sparse arrays. But this is the model, and it is very, very powerful because specifically for variants, here's what we do. And this is in one slide, the solution that we have for population genomics, and the rest is handled by TileDB, the core and universal engine. It's not customized, it's not domain specific, it is general. So as far as storage goes, there is one dimension, which is the contig, right? It's like the chromosomes, for example. Another int dimension, which is the positions within the chromosomes, and then a third dimension, which is the sample. And here, these entries are effectively entries of a single sample VCF file. This may be SNPs, and also you may have indels and CNVs. You may have GVCF ranges. We adopt the GVCF format. So you see, everything that I show you here is potentially a single VCF file, and the VCF files expand along this third dimension. And in TileDB, we are modeling only the start position of those entries as a non-empty cell, and the rest are attributes. The rest are stored, imagine, inside the cell. So the end position, ref, alt, and all of those fields are stored inside. But this is the trick, right? We are storing only the start positions of all of those entries across all samples, and we're very, very careful here because the arrays allow you to model the data or lay out the data on disk in a way that you preserve the locality across the samples. So effectively combining the flexibility of the single sample VCFs and the locality of the combined VCFs. You get both without any of the shortcomings. And now when you access, supposing that you have a rectangular query, which can be three-dimensional here. So any subcohort, any subset of contigs, any subset of positions, what I'm depicting here in blue are the results. So remember, intersections, not just inclusions. So the trick that we do is that we add these anchors over here. Again, speeding up a little bit the technical stuff, but we want to build these anchors. This is the only inflation that we have in the data set. It is minimal. It's below 1% inflation. And then we expand the range to the left by this anchor gap. So without getting into details, if we issue this query as just a generic range query to the multidimensional array engine, we are sure to get all the results and filter out whatever is not a result. And this thin layer on top of TileDB is called TileDB-VCF. And this is going to be the focus of the second part of this presentation with error. But you see, with a little trick, we reduce population genomic storage and analysis into array analysis, and everything else falls under the array realm. So all the benefits that we're building with arrays, you get it in population genomics. This is the takeaway. So a very small comment about arrays subsuming data frames. So even data frames, for example, clinical data, right, phenotypes, they can be stored in TileDB arrays as either dense arrays, which is effectively a set of vectors, one per column, or sparse arrays if you want to index those columns. Again, we have a webinar that delves into the format in detail, so I'm going to delegate all the information for further study there. But the takeaway here is that arrays can model tables extremely efficiently, so you get both the variant tables as well as the clinical tables in a single solution, and now you can fuse without wrangling and without copies. And of course, TileDB stores a lot of other things as arrays. For example, we are very big in single cell. There's going to be a different webinar for that. We do biomedical imaging. We do a lot of relevant things to healthcare. And also, of course, we store even flat files. And this is very, very relevant to you guys as well, because those BAM files and CRAM files and FASTQ files, they can be stored and managed by a proper database with TileDB. All right, so how did we build this? I have another couple of slides, which I'm going to go through very, very quickly, and then I'm going to hand it over to Dr. Kingsmore. So at the very bottom, imagine any backend. TileDB is very flexible. At the very top, imagine any API and any integration you can think of, like integrations with other SQL engines and computational frameworks like Spark and Dask, even custom integrations. We're very big in integrating even with your own tools. And we have two offerings. We have an open source offering, which is called TileDB Embedded. That's the storage engine. And then on top of this, we built the commercial offering, which is this universal data management platform, which Aaron is going to explain in detail. And then we take it to the extreme to integrate with all of those APIs and tools. So this is the software stack. Then TileDB Embedded itself comes with a lot of perks. Once again, I'm mentioning this because everything we're building for TileDB Core or TileDB Cloud gets inherited by the VCF offering automatically. We don't need to build something on top. So you're going to get all the future updates that can come, for example, from LiDAR, or they can come from imaging. We get insights and improvements from multiple domains, and then even the genomics domain gets to inherit all of those. So TileDB is very fast. It's built in C++. You can find it on GitHub. It's a columnar format, so it's very, very compressible. We have many compressors. It allows for rapid updates and data versioning. These are things, again, that will come in the future to you guys. I mean, still, the hurdle is with VCFs, but once you solve this, you're going to say, wait a second, I'm performing an update, and I want to version this, and I want to go back. So everything is built into TileDB. We have parallel ingestors, or parallel writers, parallel readers, time traveling. So all of these are inherited by TileDB-VCF. TileDB is very interoperable, as I mentioned, and very, very optimized for the cloud. And then for TileDB Cloud, remember, you're storing your BAMs and VCFs and all of those files as arrays into your own buckets on TileDB Cloud, into your own S3 buckets, for example. TileDB Cloud takes care of governance and compute, and you get to use all the tools that I mentioned. And this is built to work anywhere. Aaron is going to focus on the SaaS product, but you can deploy this on premises if you want to, and you can manage it on your own. It's serverless. You can launch Jupyter notebooks. It's geo-aware. It's secure. Once again, we have a separate webinar for all of those features. And although that webinar is generic, everything is inherited by the Genomics offering as well. And an important takeaway is that everything is an array. So even machine learning models are stored as arrays, code is stored as arrays, dashboards, and that allows you to share everything with governance. Even machine learning models, you can share them with governance on TileDB Cloud. Everything is logged and everything is monetizable. So I will stop here, and I apologize that this ran over a little bit, but with that, with huge thanks, I would like to invite Dr. Kingsmore to join us and speak about his amazing project.
Good morning, everybody. Great to be here. Thank you, Stavros. I'm going to share my screen. Let's talk about an application of this very exciting technology. Our institute, Rady Children's Institute for Genomic Medicine, are pioneers in genome-informed inpatient pediatric care. So let me explain to you why we need the TileDB solution to advance our mission and vision. So the first problem that we have tackled as an institute is shown on this slide. It's eliminating the diagnostic odyssey. So the problem, succinctly put, is that infants, if they become ill, are admitted to intensive care units, neonatal or pediatric intensive care units. We're talking about 100,000 newborns per year in the United States, or about 2.5% of newborns. About half of those babies and infants, when they're admitted, we don't know the reason for their illness. We don't know the underpinning etiology. And the problem that confronts doctors is this diagnostic odyssey. There are 7,000 known genetic diseases. How can you narrow that list in a time frame that works for a critically ill baby? And many of these diseases progress incredibly fast. And so you have minutes, hours, days, rarely weeks to actually solve this problem. What we've developed is the ability to decode the entire human genome in a matter of a day and to enable rapid diagnosis. And that in turn allows us to give specific therapy for the underpinning etiology rather than treating the symptoms. And in that manner, we can prevent unnecessary morbidity and mortality. Here's an example of a project where this was deployed at 5 children's hospitals in California. A total of 178 families received rapid genome sequencing. 43% were diagnosed with genetic diseases. This is exactly the scenario I was talking about on the prior slide. But the amazing thing is this next column, that almost a third of those babies had changes in how they were treated during their hospitalization as a result of those diagnoses. What that means is we're giving precision care, highly specific care, rather than generic care, and that's associated on average with about an 18% improvement in outcomes in these infants. Why TileDB is so relevant is that we are facing a tsunami of genomes as a community, and it's because of a coalescence of these 4 different parameters. Number one, something we've been driving has been the navy blue line, which is time to diagnosis, and now stands at 13.5 hours. That means that we can decode the genome fast enough for pretty much any sick baby. That's really important from a clinical standpoint. The black line shows another key driver, which is that the cost of decoding a genome, this is in a research setting, this is not with all of the interpretation cost, has dropped to about $650. This means that it's feasible now to really think about decoding thousands, tens of thousands, hundreds of thousands of genomes in a single project. The other 2 drivers then are the fact that our knowledge base, the number of known genetic diseases, the yellow line, has started to peak, and we're aware now of over 7,000 individual unique genetic diseases. And then lastly, a major future driver will be the number of FDA-approved gene therapies. And why this is a driver is each gene therapy requires something like a genome sequence to actually identify any patient who will subsequently get that medication. And many of these gene therapies are for these conditions in which time is very critical. So this in the future, as we go from 17 approved therapies to 170 and then 1,000, will become a major driver for the pharmaceutical industry and for physicians to deploy rapid genome sequencing more widely. This is our system that we've built over the last 6 years. It's a healthcare delivery system, and the green line in the middle is the actual genome sequencing piece and the diagnosis. But doing just that piece alone does not equip an ICU team to be able to improve outcomes in infants. It requires many more bolted on components, nor will it solve the problems that pharmaceutical companies have in terms of drug development with new genetic therapies. And this is where TileDB comes in. So we're making good inroads, not just us, but this is us. So many organizations around the world in implementing what I've just shown you. On the left are the number of sites where we offer genome sequencing across North America. It's about 72. And Project Baby Bear, Project Baby Deer, both of these Medicaid implementation projects have now resulted in policy decisions to reimburse rapid whole genome sequencing in this patient population. On the right is the number of probands, that's critically ill kids, that we decode on a monthly basis. You can see the growth there, and we expect that at this inflection point that these numbers will start to increase more logarithmically rather than linearly. So let's come back to TileDB. What is the central problem or problems that TileDB will solve for us? Well, this is the first one, and Stavros mentioned it, and for us it's a reality. It's the N+1 problem. So for 100 families and 300 or 400 genomes a month, we need to refresh our allele frequency information, and to do so in a time frame that fits our interpretation timelines. So we are endeavoring to turn results around the same day they come off the instrument. And so the N+1 problem, the ability to rerun my allele frequencies predicated on this new family's information, we have the N+1 problem in spades. When we had 500 genomes, it wasn't a problem. When we had 1,000 genomes, it wasn't a problem. We now have a problem, and that problem is going to grow as the number of background genomes grows. Currently for us, it's about 6,000 in one instance. But we want a solution that not only will solve it for the next year, but will be scalable to population size tables and formats. And so the sparse array format that Stavros mentioned is a highly optimized solution for our specific problem. And this is going to be true of anybody who aspires to decode thousands or tens of thousands of genomes and who wants to make that dynamically queryable and who needs to accrue that data periodically and therefore needs to refresh their database. With every other system that we looked at over a period of 2 or 3 years, we were finding that we were hitting the wall. It was taking us days to refresh that underpinning database of allele frequencies. That's a very specialized application, but it's one that's integrally woven into our ability to interpret new data. The second and maybe more generalizable area is that hitherto we've had a transactional interaction with genomes. And what I mean by that is they went through a diagnostic process, we reported results, and then our data was archived. But what we're now realizing is that for many of these genetic diseases, there either isn't an effective therapy today, or we don't even know enough about the natural history of that disease to be able to design a new therapy and advance its development. And so this brings us into more complex queries, real true data science, where we want to discover new insights from longitudinal admixed data that's genome, phenome outcomes. So both electronic health record data with all of its complexity and issues of some being structured and other non-structured, and then the same issue that we've had over on the left with the genome data. And some of these queries we want to ask are really computationally quite heavy. Some of them are pretty simple, but in order to be able to do this realistically, we want to make this something that we can disseminate. Our ideal platform is one that can be used by all 72 of our current partner sites. And so we envisage people pinging this data set from points all over North America. And in the future, as we integrate with other countries' data sets, being pinged from all over the world. And so it's not just a matter of running complex queries, it's a matter of running hundreds of concurrent complex queries on dramatically expanding data. Those are the end of my slides. I just wanted to very briefly give you insight into how in the real world, TileDB is allowing us now to do things that were hitherto not possible. I don't have a front end or a back end yet to show you. We're actually deploying this technology right now. It's going pretty well. Give us 6 months and we should have some fancy bells and whistles to show you. Thanks very much.
Thank you so much, Dr. Kingsmore. It's truly an honor to work alongside with you. Okay, with that, we can move on to an actual hands-on presentation, and I will hand it over to Aaron Wallen, who is going to present TileDB Cloud and specifically TileDB-VCF.
Hey, thanks, Stavros. So like Stavros mentioned, I am going to be talking about managing and analyzing genomic variant data with TileDB. And in particular, I will be focusing on TileDB-VCF. I'll just switch to the repo. So this is our application that's built on top of TileDB for working with VCFs. It's free and open source, and you can install it locally and run it anywhere you want. It offers a number of APIs, including a nice command line interface. Today, I'm going to be using the Python package and working in Jupyter notebooks that are hosted on TileDB Cloud, which provides some nice features around sharing, data governance, and compute. But I want to emphasize that it's not a requirement to use TileDB-VCF. So I'm going to cover a couple things: how to get your VCF data into TileDB, how to query it once it's there. We'll look at how to parallelize large queries so you can maximize performance, and then take a look at our framework for running analyses and building workflows that can easily scale. So let me start with my notebook here. So we need to load the Python packages. We are going to use chiefly the TileDB Python API, TileDB Cloud, and then TileDB-VCF. So I'm going to be using single sample VCF files with chromosome 1 data from phase 3 of the 1,000 Genomes project. On TileDB Cloud, we provide arrays for the more modern, high coverage version of this data that was generated by the New York Genome Center. But I'm sticking with the older data here just to keep the demo snappy. And after the webinar, we will send out an email to everyone that provides access to these notebooks, so you can start experimenting with your own data and work with any of the genomic data sets we offer in TileDB Cloud. So the first step is to create a new array to store our VCF files. So I'm defining this array URI variable here to indicate where I want to create that array. And then I'm just passing that to TileDB-VCF to define our dataset object. So this points to that URI and then opens a connection in write mode. And then we simply call the create dataset method, which actually creates the new array, which I just did there. And I will briefly open up my file browser so you can see we have this new array here. We call it demo 1kgp3. So it's there here on our local environment. So the process of ingesting the VCF files into this new array is very straightforward. I'm going to use the glob package here to identify all of the VCF files in a local subdirectory I have called batch1. So you can see this includes five different VCFs. And to actually run the ingestion, I just pass this list of file paths directly to the ingest samples function, like this. We'll run that. And each of those files is processed individually. We parse out the VCF data and ingest it losslessly into the TileDB-VCF dataset we just created. So this takes about 12 seconds to run. So we're spending about 2 to 3 seconds on each VCF file if my math is right. And if we want, we can open the array in read mode and then print out the list of samples that the array contains just to verify we have in fact ingested these 5 VCFs. So let's ingest a second batch of files into this array. So one of the most important features of TileDB is it solves painful scaling issues that are inherent to workflows that require merging large numbers of VCF files. But once a TileDB-VCF dataset is created, you can easily add new samples by just rerunning that ingest process. So here we have another batch of 5 VCF samples, and we can rerun the ingest samples function. And this new batch of samples will ingest just as quickly as the first one did, because each new sample is added to the array without touching any of the existing data. So that makes incremental updates extremely efficient and lets you avoid the really computationally expensive operations like regenerating multi-sample VCFs, which will require recreating the entire file with each new update. And while we're here, I also want to mention that with TileDB's native support for remote object stores like S3, I could have just as easily ingested these samples directly from the cloud. All I need to do is replace those local file paths with S3 URIs, and then it just works. Similarly, I could have created this array on S3 and then performed everything in the cloud. And that way you never have to worry about scaling your storage or running out of space. So now that we have seen how to create a TileDB-VCF dataset, let's look at how we can access and analyze the data. So the first thing we need to do is reopen the array, this time in read mode. And what we're doing, it kind of helps have a mental model of how we're storing this VCF data. So we're effectively modeling it as a data frame, where each row represents an individual variant identified by its chromosome, chromosomal position, and then sample of origin. So this lets us quickly slice out specific variants by querying for a particular genomic region or a specific sample or group of samples or both. And all the queries from Python are performed using the read method, which is what we're doing here. And in this case, I'm just asking for all of the variants that overlap gene mTOR1, so the genomic coordinates for mTOR1 for this particular sample. And I'm also indicating which VCF attributes I want to include in the results. Since TileDB is a columnar format, it only touches the files that contain the attributes I've requested. So we don't have to waste time retrieving and returning every single info and format attribute. I can just pull out what's needed for my analysis, which can provide a real performance boost, especially when you're working with remote data. When I run the query, we're in Python, I always get the results back either as a pi-arrow table or a pandas data frame, which are excellent starting points for working on your downstream analyses in Python. So that demonstrates sort of the basic mechanics of how to construct and execute these queries. So now I'm going to start talking about scaling, which is often where things get a little painful with genomic analyses. But with TileDB, we support fully parallel I/O, so queries can be distributed across as many cores as you have access to, with each core processing a different chunk of the data. And TileDB-VCF provides a simple interface to set up distributed queries like this by allowing you to easily split the queries by region or by sample, or both, into discrete partitions. So as an example of this, I'm going to perform the same query I ran above, but now I'm going to run it across all 10 samples and split those 10 samples into 2 partitions. So to do that, I need to set the sample partition argument here when I reopen the data set. And when I run this again, TileDB-VCF will automatically limit the query to include only the first half of samples in this data set. And then I could query the second partition or the second half of these samples by, again, reopening the data set and then change my sample partition to the second partition, which is 1 here. Reopen that, run the query, and then you can see we're getting a different subset of samples now in our results. So now we can do the exact same thing with query regions, or like I said, we can do it, we can partition both samples and regions, so you can break up really large queries into block-wise chunks that, again, could all be executed in parallel. So let's take a look at how to do that in the next section. So TileDB-VCF, with it provides a couple of options for users to leverage these partitioning features to run queries in parallel. So we offer integrations with frameworks like Dask for parallel computing in Python, as well as Apache Spark. So if you already have those services set up in-house, then TileDB-VCF will slip into your existing workflow quite easily. But today I'm going to use TileDB Cloud to build my pipelines. And if you're new to TileDB Cloud, Seth Shellnut, our CTO, provided a really great introduction in a previous webinar, so I highly recommend checking that out. I won't spend too much time introducing any of those features, so it's worth taking a look. But specifically, I'm going to be using the compute features. So TileDB Cloud pipelines are built around user-defined functions, or UDFs. So each step in your pipeline is one UDF, which is shipped to a node and executed with the arguments you define before returning the results. So this is a really flexible framework on top of which you can build any kind of data processing pipeline. So I'm going to start with a simple example here that queries the full 1,000 genomes data set for a particular variant. The first thing I need to do is import the TileDB Cloud package. So this is what allows me to create delayed versions of normal functions that are executed serverlessly. So here I'm defining the UDF I want to run. It's a very simple function. It just opens a TileDB-VCF data set and then it queries it. One notable modification here is I'm returning the results as an arrow table rather than a pandas data frame, just because the arrow format is a little more efficient to serialize and return. But otherwise, there's not much to this UDF. We keep it very simple. So to create a delayed instance of this function with the parameters needed to perform the query, I will define the parameters I want to pass. So I need to tell it what's the array we're querying, what are the VCF attributes we're pulling out, which samples are we querying in which regions. And then I'm passing it to the delayed function and then telling it, this is the UDF we're running, VCF query, and then here's the parameters. So that's the process. And this samples argument includes a list of 100 samples. For this first step, I'm just limiting it to the first 10. So we created that and nothing happens, because to actually execute the function, we need to call the compute method here. So that's where the delayed comes from. So if I run that, it's actually doing what we said earlier. It's serializing that function. It's sending it up to a node in TileDB Cloud where it's executed. And the whole process takes about 12 seconds here. But as a user, I don't have to manage any infrastructure. Everything happens automatically for me, and I only pay for the compute that I actually used. And again, we're getting the results back in an arrow table here that I converted to a pandas data frame. So now we have the groundwork to start running really large queries in parallel and on different nodes that each return a different slice of the data. To really do this at scale, though, we're going to need one more UDF, which will take those different results and then combine them into a single table that's returned to me in the notebook. So that's what we're doing here with this combined results function. And to perform our full query, we have to arrange these two UDFs into a task graph. So what we're doing here is we're splitting up the 100 samples we want to query into 10 partitions that will run in parallel. So we're looping over this 10 times and creating a delayed instance of that VCF query with each pass. The parameters are identical except for the sample partition index here, which is what we're incrementing each time through. And then each instance of this UDF will be saved in this delayed results list. So this is going to be a list of delayed functions. We will take that and then pass it to the combined results UDF, which will, as it's named, take the results and then combine them into a single table. And it's always easier, I think, to mentally digest this when you can visualize it. So let me create that task graph. Now you can see what it looks like. So each node here represents a single UDF. So the first row, we have 10 nodes for each of the 10 sample partitions. And they're all connected to the root node here, which combines the results. So that's our task graph. Like before, to actually execute this, we need to call the compute method here, and that actually kicks things off. And so the graph updates in real time to indicate which nodes are running, which ones are finished, so you can easily track the progress of your pipeline. And again, our 10 queries, they're all running in parallel. Those results are all sent back to me. And we'll convert it to pandas here. So now we can continue on with our analysis, in the notebook. So anything downstream we want to do, we have our results here. So by storing the VCFs in TileDB, we have the ability to efficiently slice the variant data. We can handle large queries by splitting them up into the sample and region partitions. And with TileDB Cloud, you have the ability to spin up as many nodes as you need to execute those queries in parallel. But where this gets really interesting, I think, is we can build on this approach to create scalable pipelines that perform common analyses like a genome-wide analysis. So I am going to switch to another notebook and look at an example of that. So this is another notebook we'll be publishing on TileDB Cloud. And like I said, it covers how to implement a basic genome-wide analysis using TileDB-VCF. So one of the goals of TileDB Cloud is to make it really easy to share with your team and collaborate on research. So I have the notebook here, but it gets a special URL on TileDB Cloud that looks like this. So every notebook will get an overview page where you can provide a README style description. It supports full markdown. You can add a license. You can see a preview of the notebook. So when you share it with someone, they can see exactly what's inside without having to actually spin up a Jupyter environment. And so if I wanted to give everyone in, say, the TileDB company access to this notebook, I just hit the sharing tab, hit the share button, and then we have an organization for the company that is, it's TileDB Inc. We'll go with that. And I'm going to give everyone read-only permission. And now everyone at the company who has an account and is part of this organization would be able to access this notebook in the notebook section here. And importantly, when you're sharing notebooks, you get a full activity log, so you can always see who accessed it and when and what was done. And right now it's kind of lonely, it's just me, but hopefully that will change once we publish this and everyone has a chance to work on it. So all of these sharing and management features that we just looked at for the notebooks, they're provided for every one of the assets listed here on the left. So arrays, notebooks, UDFs, dashboards, machine learning models, they all get the same set of features because, as Stavros mentioned, underneath the hood, they are all stored as TileDB arrays. So all of this functionality is just inherited automatically, and everything can be managed using the same set of APIs. So if someone shares a notebook with you, you can come here to the overview page, just hit the launch button, and now you're working on the same analysis, you're working with the same code, the same data sets. There's no need to duplicate anything. So I already have it opened here, so I don't need to launch an instance. And as I was saying, the goal of this notebook is to construct a rudimentary GWAS here. And we're implementing everything ourselves in Python just to illustrate the process. But this basic approach can be easily adapted and customized to use whatever tool or algorithms you need. So I'll start off by loading in our packages. We're using the same suite of TileDB packages that we used before. We're defining a couple variables here. So again, we're working with the phase 3 1,000 genomes data set. These are the attributes we are going to pull out from that data set when we run the query. And because there's no phenotypic data available for these samples, I'm just creating a dummy trait here for us to analyze. So to get started, I'm calling this function I wrote called genome windows, which divides a genome into discrete bins or windows that will let us parallelize our genome-wide analysis. So I will run this here. And for this demo, I'm just focusing on chromosome 20 and then splitting it up into 100 kb regions. And throughout this notebook, I'm going to use a number of different UDFs, but I'm not actually defining them here like we did in the last notebooks. There's no source code for them. Instead, they're all registered on TileDB Cloud. And let's switch there. So just like the notebooks, they get this nice overview page. So I'm using the README here to document the function's parameters and its output. We can peek at the source code if we want. We can look at the activity to see who's using the UDF. And we can share it with anyone we want. We can make it public. So we get all of those same features as before for this UDF. So easily shareable code. It's great for reproducibility. It makes it really convenient to reuse these functions by just calling the URI like I did here. So the actual GWAS, the association analysis, is broken up into these 4 steps. Each one is implemented in a separate UDF. So first, we are executing our TileDB-VCF query. This is very similar to the VCF query UDF we ran in the last notebook. In step 2, we are going to filter those variants. So this UDF will actually calculate allele counts and minor allele frequencies so we can remove variants that don't meet our filtering criteria. And then step 3 is where we actually perform the association analysis. So in this case, we're using a Fisher's exact test for each variant and then returning the summary stats in a pandas data frame. So you can see for each variant, we'll get an odds ratio and a p-value. And the last step, like before, is we need to combine those individual results into a single table. So those are the UDFs. Each of those is registered on TileDB Cloud. So anyone can access them, anyone can look at the source code, and you could use them in your own analyses if you need to. To assemble these steps into a task graph that we can run serverlessly, it's very similar to what we did before, except now we have these multiple steps with dependencies between them. So you can see when we build the task graph, we're looping through partitions just like we did before. So we're going to have one query for each of these region partitions. The results are stored in the delayed queries list. And then the results from delayed queries will be passed to the input of our delayed filter step. The output from the delayed filtering step is then passed to the delayed GWAS step. And let me run this. Again, it's always easier to visualize this thing. So this is what the final task graph looks like. And you can see we have 3 main stages. So the first row of nodes corresponds to the queries that are run in parallel. Second row corresponds to the filtering step, and the GWAS step, and then everything is combined at the end. So the whole thing takes about 2 minutes or so to complete, so I won't run it now, but again, we're going to share these notebooks so you'll be able to experiment on your own. But the real take home here is any type of genome-wide analysis can be implemented using this approach where you're dividing the genome into partitions and then analyzing those chunks in parallel. So I have one more demo I would like to cover. Let me switch over to this last notebook here. So this notebook walks through a clinical genomics type scenario in which you would like to identify how many patients with a particular condition carry a coding variant in a gene of interest. So to perform that kind of query, we need to start bringing in external data sets, not just the variant data. So at minimum, we're going to need patient genomic variant data. We're going to need genomic annotations and variant annotations, and then information about sample phenotypes or metadata, whatever data you have collected for your samples. So we'll look at each of these different data types one by one, and then we're going to see how we can integrate everything into a single task graph to run this query. So I will start with the variant data here. Sorry, I flew by the variables. So in this notebook, we are actually using the high coverage version of the 1,000 genomes data set. So this is the more modern resequencing effort that was performed by the New York Genome Center. This array is publicly available on TileDB Cloud. So the whole thing is about 11 terabytes, which is a drop from the original GVCF files, which were about 15 terabytes total. And we created this array by downloading all of those GVCFs from the FTP server, copied them over to S3, and then ingested batches of samples in parallel with TileDB-VCF. So we've talked about how our queries can be parallelized, but the same is true for writes as well. And in fact, we have a notebook available that demonstrates how to perform a parallelized VCF ingestion on TileDB Cloud. So this is a fairly large data set, but what's nice is we can slice it directly from the cloud. There's no need to download the entire thing to run an analysis. And again, this is publicly available, so you're very welcome to use it in your own work. And of course, we thank the New York Genome Center team for generating it and allowing us to make it available here. And I'll find my notebook, there it is. So that's the array we're using in this example. So we've already seen how to query variant data, so I will skip that part of the notebook and move over to the gene annotations, which is this section. So we want to be able to limit our variant query to only include coding regions for a specific gene of interest. So to do that, we created TileDB arrays that contain gene and exon annotations that were imported directly from Ensemble. And now we can easily query those directly using Python, which is what we're using here, or any one of our APIs. The array is indexed by gene symbol, so we just need to specify what our gene of interest is, and then it returns the relevant annotation data in another Pandas data frame. For the full query we're going to perform in a task graph, we want to return bed file formatted regions for the exons. So we actually accomplished this pretty easily with SQL. And it's nice that we have the option to use whatever our preferred tool is, or whatever the right tool is for the job when working with TileDB arrays. So that's what I'm doing here in SQL, but rather than running it directly, I am creating a delayed SQL statement, which is sort of the SQL analog of our delayed UDS. And this is what allows us to include it in a task graph. So for the sample annotations, they were provided as a CSV file that is easily converted to a TileDB sparse array with the Python package, which has a from CSV method. And for the query, I want to filter samples to include only females that come from a specific population, because we don't have a lot of other sample metadata to work with here. So again, we will create another one of these delayed SQL statements that will perform that filtering step for us. So to perform the full query with all three of these data sources, we're going to create another task graph. So we've already created the SQL queries for the sample and ensemble queries. To include the VCF query, We have another VCF querying UDF. That's actually the same one we used in the GWAS demo, so we don't need to go over that. We just included it here as part of the tutorial. And then again, we have our old friend that combines those results. So the process of assembling this task graph is, again, very similar to what we've seen before. We're splitting our variant queries into, I think, 5 partitions here that are run in parallel, and then combining those into one final step. So this is where we pull it all together and visualize the task graph. So the first 2 tasks here correspond to the delayed SQL statements that I think, yeah, this is the SQL statement that filters the regions, creates those bed file genomic coordinates for DRD2's exons. This is the SQL statement that filters the sample data. There's no dependency between those 2 things, so they are automatically run in parallel. And then those results are returned as inputs for the 1,000 genome variant queries, right? So that's where the sample input comes from and the query regions. And then those are all run in parallel before we return the final result here. So I ran this before, but I will start it again. So this kicks off the process. So we can see those 2 SQL statements have started already. And we can monitor the progress here, but we can also look at all of the tasks we have running on TileDB Cloud with this task view. So you can see right now we have those 5 variant queries that are all running. You can click on any one of these. You'll see the source code that was actually run in the UDF. Any errors or logs you want to print would all be available there. So it makes it very easy to track to see exactly what was happening in your task graph. You can also see how much each of these individual steps costs. And once that's done, again, we get our pandas data frame with our filtered results. So these are the variants that overlapped DRD2 exons, and then we could continue on with our analysis. So that was the last demo I had. Looking forward to taking your questions about what I've shown here. But for now, I will pass it back to Stavros.
Thank you very much, Aaron. So we have a lot of great questions. I'm going to spend 1 minute on our future work, and then we will answer every single question. So going back to my slides, just for a second. There you go. Okay, so work in progress, or in other words, and this is actually why I want to present this slide, why you should depart from file formats and specialized solutions. The point I'm making here is that we're working on some amazing things that may sound to you irrelevant to genomics, and yet they're super relevant to the performance you're going to be seeing in several features down the road. For example, one of the things that we're working on is RLE compression for strings. So RLE compression specifically for sample names. This is going to give a huge compression ratio for the sample field that we have, but also it will allow us to compute directly on the compressed data. This is important because it's going to save a lot of computations internally. It is going to save a lot of memory budget. And when we perform distributed computations like the ones that Aaron showed you, it's not just the CPU that we're optimizing for memory as well, because the more memory, or the other way around, the less memory each of the tasks consumes, the more tasks we can spin up on a node. So this is very, very important. And this is coming up. Again, this is generic. It has nothing to do with TileDB-VCF, but TileDB-VCF builds upon TileDB embedded. This is going to be in TileDB embedded, and TileDB-VCF will completely transparently inherit all of that. Another thing that we're doing is that we're very big in pushing down several computations from the tools we're integrating with. There is a question about Hail, and we're going to respond to that in a bit. But not only for Hail, but also for the SQL engines, for generic Spark that we're integrating with, and so many other geospatial tools. You can't imagine how common the operations are. You know, the filters, the aggregates, they're the same operations, whether it is VCF or LiDAR. So we are taking those computations, so we're going into the code of those tools that we're integrating with, and we're pushing this down to TileDB embedded to be computed closer to the data, so that we avoid copies. We do it in a multi-threaded manner, in a vectorized manner, and so on and so forth. Again, this will be inherited by the VCF offering. Another thing that we're doing on TileDB Cloud is fine-grained access policies. So far, Aaron showed you how to share, for example, arrays, but in their entirety, so an entire population genomics data set. Very soon, you're going to be able to share on the level of a variant as well, and actually of a variant field. So we're going that deep down and that fine-grained in terms of access policies. And finally, we're performing massive performance optimizations. Every day we have dedicated teams to work specifically on performance. So once again, all of this is going to be inherited by TileDB-VCF, and this is the reason why I always suggest to all the domains, this goes way beyond the genomics domain, that you should not be focusing on specialized file formats. You know, focus on defining APIs, invite other companies and other engines to be built around those APIs, and then you should be able to move from one engine to another just by linking to a different library. You should not be focusing on file formats. You should not be focusing on specialized tools working on those formats. So with that, the presentation is complete, and we're super happy to answer the questions. We have some amazing questions. We're going to answer them one by one.
Awesome. Thanks. All right. First one. How does TileDB compare to Hail in terms of features, performance?
Yeah, also, I would like to mention that we have Seth Shelnut on the webinar as a panelist, our CTO, and he has been very involved with a lot of different things, of course, but also with TileDB-VCF. So Seth, please feel free to chime in any questions. I will take this one, but please feel free to chime in at any point. So first of all, we integrate with Hail, right? Hail is based on Spark. We integrate with it. We integrate with Spark directly as well. So it's not a direct comparison. It's kind of complementary. We're solving the data management problem, and then Hail has a lot of nice features around annotations and many other things. And you can still inherit them, and you can still have them. The biggest problem that we saw, and that the problem we're solving, is that Hail ingests a combined VCF file. There are some experimental plugins and code paths to ingest single sample VCFs, but the main thing is the combined VCF. And what I told you in the beginning is that the combined VCF file is the problem to begin with. So what we're saying is, don't combine the VCF a priori, keep the data into the single sample form in TileDB, and then we will export on demand, right, to Hail's internal structures, this combined VCF format. So that's the kind of integration we're doing, and you're gonna see a lot of updates on this soon. So no reason to choose, one way or another. What we're offering you is just extra flexibility, because a lot of the features built on Spark and built with Hail, you can build on your own with UDFs and task graphs the way Aaron has showed you. So extreme interoperability and extreme flexibility.
Okay, next one. Is there any support in TileDB for more complex queries, e.g., oops, sorry, let me start over. I'm reading the wrong question. Someone dismissed it and it popped down. My bad. VCF/BCF is great thanks to TabX indexing from HTSLib for querying by region. I see TileDB has similar region querying. How does performance compare to HTSLib on region querying?
Yeah, so again, I'm going back to the original problem. Slicing a region from any of the files, single sample or combined, is fine. with HT-Sleep. And I agree, it's because of the topics indexing, right? The problem is that if you keep the single sample VCFs as they are, you need to do this slicing on each of the files, and this latency is gonna kill you across 1 million files. And then for the combined VCF file, again, I mentioned this, that accessing is very fast. Like you collocate the variants, the sample information for each variant. So if you're looking for a variant, you can find it fast, and then the rest of the information across samples is collocated so you can scan it very efficiently. That's not the problem. The problem is creating those files. And another problem is that HT-Sleep was not built having the cloud in mind. So it's not easy to just extend it and work on S3 very, very efficiently. TileDB uses R trees. So it's rapid in terms of slicing. And it goes beyond that. In addition to having the R trees for each of the right batch to slice on variants and samples, we have this time versioning and time traveling functionality so that you can keep on adding batches of samples like Aaron showed you. So, and we have an extra index layer on those fragments so that we get only the relevant ones, be very considerate about the listing requests on S3, being very particular about the any kind of REST request on S3. So TileDB takes it to the extreme when it comes to performance, but versus HT-Sleep, TileDB is rapid, like due to the R trees. So that's what we do. It's a multi-dimensional version of the topics indexing of HT-Sleep.
All right. Sorry, Seth, I thought you were about to chime in. So, okay, next one. Is there any support in TileDB for more complex queries, e.g. querying on functional annotation and genotype, e.g. get all samples in my TileDB with a homozygous pathogenic variant in any known disease gene?
Aaron, you can take this one.
Yeah, so some of this is coming from the push down filters that Stavros alluded to earlier. So any of those info or format attributes in the original VCF file are ingested into TileDB. You can think of those as sort of columns in your data frame. And you're going to have the ability to include query conditions on your query. So, give me all the variants in this region, but only if the read depth is greater than 10, for example. As far as the external annotations, something like VEP data, the workflow we've seen with customers is typically that data comes in a separate annotation style VCF file. We will ingest that into a separate array and then perform the kind of workflow I showed in that last notebook, where we're in parallel slicing the variants from the sample data, grabbing those same regions from the VEP array, and then performing a join in SQL or Python to implement the filters to grab, only the particular mutation, a nonsense mutation, for example. But in the future, we are looking to take advantage of a new feature that was added to TileDB Core, which is the ability to evolve the schema, add new attributes to an existing array. So that way we can run a UDF that performs something like VEP, stores the results in the existing VCF data set.
Cool. Okay, thanks, Aaron. All right, so the next one, one more. Have you tested TileDB on mega large projects, e.g. the exome data coming from UK Biobank? UKB have had to resort to making their latest exome data available only on an online research platform, i.e. you have to bring your tools to the data rather than vice versa.
Yep, we're very familiar with the platform, and we can discuss offline anytime. But we have our Helix friends on the webinar, so I got specific permission to talk about this. The Helix use case is at least as big as UK Biobanks. Actually, it's bigger because the samples keep on piling up. So we have battle tested this for years at the hundreds of terabytes of scale, and TileDB is built to perform at hundreds of petabytes scale. In order because this goes beyond the scope of this webinar, we do have a webinar on the TileDB embedded format where we explain how this works very, very well on cloud object stores as well. And it's quite evident how TileDB can handle this kind of scale. It's because of the format and because of the way we architect the whole storage engine. That's the reason we can do it. Very happy to schedule a call offline anytime.
Thanks. Okay, what about aggregate statistics, e.g. allele frequency, HWE? Any way to query by AF?
Yeah, Aaron, you can take this one. Similar to the previous one.
Yeah, it is. It is very similar, and similar to what I showed in the GWAS notebook, right? As part of that task graph in step 2, I actually had to calculate the allele count, allele number, and the allele frequency, so we could filter variance based on minor allele frequency. So I didn't store those anywhere. I just used them in a filter and then, moved on. But what we could have done was taken those results after calculating them and then stored them in a separate TileDB array that was just variant centric, since those are variant level stats that are calculated over an entire population. So it's definitely possible that would be the workflow I would use. Use that GWAS notebook as a starting point, but add a step that actually ingests those results into a new TileDB array that's indexed by the variant positions and stores each statistic in its own attribute.
Thanks, Aaron. Okay, all right, next one. Can you query and retrieve the results back and not have sample as an index, e.g. variants don't get duplicated if they exist in multiple samples?
Yeah, I'll try to take a stab at this if I understand it correctly. So of course first of all, you can slice for all samples, not specific samples if you want to, but I do understand at least part of the question that has to do with replicating the sample name, for example, for all the variants. And this is where we already compress. So that gets compressed very, very well. But this is very relevant to the work in progress I mentioned. We are going to be performing RLE compression on the sample names. And again, we lay out the data in a way that we collocate the variants across the samples. So this is going to allow us to do, actually, I also need to mention dictionary compression. Both of them will work depending on the use case. And then we will be able first to not report those sample names for a specific variant. And we will also be able to operate on them without decompressing them. So a lot of cool optimizations around this area. I don't know if Seth or Aaron had something else to chime in here.
Yeah, part of our work that we're doing too with some of the combined export capabilities is the ability to get the results in memory in that combined variant-centric format. So besides just exporting to a combined VCF file singularly or distributed, but also in Spark or Python, or potentially even a CSV style export on command line, getting it back in that variant-centric format. So both of those, the variant-centric and then the compression, solve this issue and provide a lot of flexibility in how you actually want to query and handle this data.
Okay, thanks. Aaron, were you all set there? Okay, cool.
No, I agree with Seth.
All right, sorry. All right, next one. Thanks. There's actually a question and then a subsequent comment on it, so I'll read both, I guess. So do you recommend downstream annotations such as pathogenicity and intersections with variant disease databases to be done after the ingest into TileDB, or should everything have to be done on the VCFs themselves? And then someone else posted a follow-up. If you can do it afterwards, that would be great, as you can save yourself from annotation work by only working on the new variants identified in the sample rather than re-annotate every variant.
Yeah, so generally the workflows we've used with customers who have a use case like that is they do store the annotations in a separate data set. And the reason for that is, if those come from a public resource, those are updated on some kind of schedule. You don't want to have to touch your giant array of variant data. You can just refresh the annotations as needed and keep those discrete. And because we can parallelize the queries for those two arrays, there's really no performance hit to store them in separate locations.
Yeah, and I would like to add that we will store all this data as tables modeled as arrays, so that you can take advantage of pretty much everything. The versioning, the logging, the access control, and of course, no data wrangling when you fuse, when you effectively join this data.
Thanks. Next one, how to adapt the different attributes in VCF files from different sources. How flexible is it to change the schema? E.g., can we change/add dimensions after creating array? And then there's a couple more. How to manage arrays, related arrays, VCF and expression data in group, security, AWS or premium. How to inject very big Excel files into array in batch. So a few ones there. Let me know if you need to repeat.
We can respond to each of them separately. So Aaron, you can take this one if you like. How to adapt to the different attributes in VCF files from different sources?
Yeah, I think this is a similar answer to the last question, right? We're getting these kind of external annotations from different sources, and you're storing them as tables in arrays. So that way you can still use all of the various TileDB APIs to access and query this data. And if you want, you can incorporate those into task graphs like we did earlier. But we have taken a number of different types of genomic annotation data and ingested them into TileDB arrays very easily. So we have data from Ensemble, as we showed. We have data from ClinVar and the VCFs they provide. We have ingested VEP data that was generated from the 1,000 genomes data set. So these are all stored in independent arrays that you can then leverage for your queries.
Thank you, Aaron. And perhaps Seth can respond to the question around the schema changes.
Yeah, so we do have support in TileDB for adding or removing columns. We call them attributes. So that will be coming to TileDB-VCF very shortly. We have a branch with that now. We're continuing to expand upon that support, eventually being able to change data types, to change nullability, and different things there. It's important to note that we already have support in TileDB-VCF for ingesting VCF records that might have different fields or different headers directly. So you don't have to have necessarily a common set of fields among all your VCFs. If you have different headers, we know, especially as your data changes over time, QC pipelines are modified. That is supported today. What is coming soon is the ability to change the fields that are materialized and stored on disk, which is a way of gaining performance for very commonly queried info or format fields.
Great. I can take the next one, how to manage arrays and anything around data management. So we're hoping that this was answered during Aaron's presentation. So everything gets registered there. You own the data. You convert the data into the TileDB format in your own S3 buckets. You create one role just for TileDB, giving it either read access or read write on Amazon, for example, on AWS. You register that key to TileDB Cloud, and then from that point onwards, the governance is done by TileDB Cloud at global scale, and we take extreme measures for security. We have certificates. We're being audited by external parties. We can provide all this information. So we always keep the key encrypted. It cannot be touched by any user's code. So all the data management is what we offer with the TileDB Cloud product. And finally, how to inject very big Excel files. We have an ingester from CSV. So if the files can be exported in CSV, you can do that today. As I mentioned in my presentation, data frames are subsumed by arrays, and you have a lot of flexibility on which column to index as a dimension so that you expedite slicing. Seth, would you like to add anything to that?
The only thing to add there is, we're happy to discuss more specifically what your use case for the Excel spreadsheet data is. Because one common thing that we've seen is a lot of people store metadata in Excel spreadsheets, and you, in addition to storing the data itself in TileDB form in a data frame, you might also be able to store that metadata in TileDB array metadata right with the data itself. Many times people have these Excel spreadsheets of, this VCF file is from this run, and some sample data. And we have a lot of ways to handle that. So there's a lot of flexibility here.
Thanks. Okay, is TileDB-VCF supported on Windows now?
I can go ahead and take that. We don't offer pre-built binaries for Windows today, but it should be fully possible to run TileDB-VCF on Windows. We're not using anything specific for Linux or POSIX. Everything we do is always cross-platform in mind. If you have a use case for Windows, we're happy to talk about it, find out your exact platform versioning, and even provide precompiled binaries there if needed.
Thanks. Do you have integrations with GA4GH APIs or any plans to implement them if not?
Yeah, I can attempt to respond to that. I'm very familiar with GA4GH. That was definitely part of the work we were doing with Intel Health and Life Sciences and the Broad Institute when I was working at MIT and Intel Labs. I think first of all, I think it's a great effort. This is what the domain should be doing: define the APIs and then invite implementations so that the scientists, bioinformaticians, right? They focus on the science, and then data engineering, databases, data management can focus on doing that very, very well, right? So this is, in my opinion, this is what we should be doing. The only note, we're very happy to integrate with GA4GH if we have a particular user, a customer that prompts us to do that. Very happy to do that. We have been doing tons of integrations. Very happy to do that too. The only note that I have, this is just a general note around designing APIs. The APIs must be designed in collaboration with those people that are building these database systems. I'll give you an example, not for GA4GH, for anything. It could be for ODBC connectors, for example, in databases. We are dealing with trillions of variants, right? We're dealing with petabyte scale data. If an API is asking this data variant by variant, the performance is going to be destroyed, right? So you may say, but yeah, this is a clean API. I want a specific variant and I want to iterate over the samples or something like that. And you make a call for every variant, for every sample, then the performance is going to be absolutely impractical. So extra care when defining any kind of API, not only in genomics, but anywhere, it has to be done in collaboration with those people that have at least a proof of concept, right? Like build a solution that implements it in a real application, right? Petabyte scale. And then you will be able to refine those APIs. But from a philosophical standpoint, my opinion is that this is exactly the direction we should be taking. Define the API, make it as easy and as performant as possible, and then let other people build anything below that in terms of infrastructure.
Thanks. Okay, if I create an array and then you make an update to improve storage, how do I apply that update to my existing array?
Yeah, this is definitely a great question, and this is very relevant to the RLE, performance improvement that we want to do. So we are building utilities. We have one that is being merged, or Seth, remind me, probably it's already merged, where you can update a version and you can configure it to do different things, because if it rewrites the whole array, obviously it is going to be expensive. But the good news about the format of TileDB is that we can do incremental changes, for example, on a fragment basis, without interrupting the normal operation of the whole database. You will be able to read with consistency and everything, while this reformatting is happening incrementally in the background. And you can even choose which portions of your array you will want this update to take place. And so we know exactly what you're talking about here, and we are taking all the proper steps in order to be able to make it very practical for you guys.
Thanks. Have the TileDB multi-array datasets used in any ML example applications? Invariant invariant prioritization, for example. So, have the multi-array datasets used in any ML applications invariant prioritization, for example?
So we don't have an application specifically that runs such an algorithm, but what we build with TileDB, and I would like also Seth to chime in on that. What we build with TileDB Cloud is an infrastructure solution, right? It's actually a combination of an end-to-end solution for population genomic analysis and an infrastructure so that you can build anything on top. And a big portion of this, a big aspect of this is managing machine learning models, right? Training the machine learning models and managing and servicing the machine learning models as part of the data management platform, right? Machine learning in our eyes is a data management problem as well. So it has to be part of this integral infrastructure. So we give you all the tools to be able to design and run algorithms like that. We integrate with popular machine learning tools like PyTorch, scikit-learn, TensorFlow, so that you have the whole toolkit in order to be able to build your own solutions on top. And I don't know if Seth has any more ideas on this.
Yeah, I just want to start off by saying too, we do have a whole team dedicated to ML here at TileDB. So we take it very seriously, and like with any of our integrations with third-party tools, we take performance very seriously. So we have a number of low-level integrations with like the dataset APIs of like TensorFlow and PyTorch and scikit-learn to allow you to do high-performance distributed training, high-performance distributed predictions, and then of course, as Stavros mentioned, also storage of the models themselves as TileDB arrays. So our integrations with ML go beyond just TileDB-VCF. We support a number of different use cases there. So we have a lot of flexibility in building out that type of framework.
Thank you. Okay, can you partially turn off logging for use of notebooks /UDFs? E.g., you have some clients that don't want people to see what they're doing, but you want to track usage for the rest of your clients.
Yeah, I'll just start by saying that you have full flexibility to do whatever you want. We control everything on TileDB Cloud, but also for certain things that you own, only you and your organization is able to see the activity for, not the rest of the world. But with that, I would like Seth to explain everything, because Seth and his team built this functionality in TileDB Cloud. We control everything. It's very easy to have different toggles so that you can hide anything you like, or if you want to reveal anything you like.
Yeah, thanks, Stavros. And to clarify, when Stavros says we control everything, it really means that we have the knobs to do everything. It's all done through TileDB Cloud and the software that we've built out. So we have a lot of flexibility here. And with TileDB Cloud, you can run our SaaS product, but also we have the on-prem enterprise edition, where you have even more control over all the different knobs that can be turned there. But first and foremost, TileDB Cloud is built around role-based permissions. So we've showed you some things in the cloud today, in the UI today. But depending on the role you have in an organization, whether you're part of it, whether you're a third party, whether you're just an anonymous person on the internet, varies drastically in what you can see and the information that's presented to you. And what we have in the user interface today is the basis of everything there. But even programmatically, you can access some of this data and get the very low level details and turn those knobs. So if you have your own compliance requirements that you need about, some data has to be logged like this, other data like that. We provide a lot of flexibility in the APIs, so you can pull that back into your own system so that you can meet any compliance requirements you have. And so you can toggle that tracking and logging on your own data sets or your own notebooks or UDFs at that granularity that you need. So we're very happy to engage and talk a little bit more about your compliance requirements there. Everybody's got special stuff, especially in the healthcare world. We know there's a lot of variation. So we've built a lot of flexibility. So we're happy to talk more about that offline.
Awesome. Thank you so much for all the questions. That was the last one. Please feel free to engage with us offline. We're very accessible, very responsive. Please do reach out, schedule calls with us. We're very happy to have a technical deep dive with you. We are going to be posting a lot of webinars, a lot of notebooks, examples, tutorials throughout the next couple of months. But still, if you want to see some preemptive peek, reach out to us.